Think of your body as a high-performance electric vehicle. If you are constantly exhausted despite consuming adequate calories, the problem isn't a lack of fuel. The deficit lies in your cellular engines, the mitochondria. This specific presentation of chronic fatigue traces directly back to a severed communication network in your body, known as the gut-mitochondria axis. This chart tracks cellular energy production. When the microbiome environment is severely depleted, mitochondrial ATP generation, the actual electrical currency your cells run on, can plummet by up to 40%. Under healthy baseline conditions, the trillions of microbes in your gastrointestinal tract act as biological signalers. They ferment dietary fiber into chemical metabolites that travel through your bloodstream to your cells. These specific microbial signals provide the exact instructions that tell your mitochondrial engines to either multiply and produce energy, a state called fusion, or to fragment and conserve resources, known as fission. Your daily capacity to generate physical energy is dictated entirely by the integrity of this internal microbial cellular partnership. The primary saboteur severing this internal power cord is glyphosate, the active chemical ingredient in modern agricultural herbicides. Its integration into the global food supply has reached a saturation point. Continuous dietary exposure has turned a targeted agricultural tool into a permanent biological stressor on the human metabolism. Standard rest cannot resolve this cellular deficit. Reversing this metabolic decay requires a clinical intervention that addresses both the internal biology of the gut and the external biophysics of the tissue. We accomplish this across two vectors: chemically rebuilding the microbial grid using a methodology called the RISE framework, and physically jump-starting ATP production through targeted photobiomodulation. Saving the compromised cell requires a coordinated rescue operation deployed simultaneously from inside the gut and outside the body. Regulatory bodies historically classify glyphosate as safe for humans based on a single biochemical detail. Human cells do not possess the shikimate pathway, which is the exact metabolic route the herbicide disables to kill weeds. Look at this diagram of the shikimate pathway. While human cells bypass this biochemical route, approximately 54% of the core bacterial species in the human gut depend entirely on this specific EPSPS enzyme for survival. Glyphosate acts as a glycine analog, meaning its molecular structure perfectly mimics the amino acid glycine. Because of this mimicry, the human body mistakenly incorporates the herbicide into its own protein synthesis, aggressively corrupting cellular construction during tissue repair. Back in the microbiome, this precise chemical targeting selectively eradicates highly sensitive keystone bacterial species like Lactobacillus and Bifidobacterium. This targeted eradication leaves a biological void. Opportunistic pathogens with a natural resistance to the chemical, specifically strains like Salmonella and Clostridia, quickly proliferate to colonize the empty space. Furthermore, the pure chemical isolate represents only a fraction of the threat. Commercial herbicide formulations utilize highly specialized coformulants and adjuvants engineered strictly to break down rigid plant cell walls. Molecular toxicology data indicates that these specific coformulants are up to 1,000 times more toxic to human cellular barriers than the isolated active ingredient. This combination of corrupted protein synthesis, of barrier destruction, and unchecked pathogenic overgrowth locks the metabolism into a state of decay. Natural recovery cannot occur without deliberate clinical intervention. To re-engineer this compromised internal ecosystem, we deploy a structured clinical strategy known as the RISE methodology: reset, identify, support, and excel. Phase 1 is the reset. This mandates the absolute cessation of compromised dietary inputs, physically cutting off the primary source of environmental stress. Simply stopping exposure leaves residual chemicals. We introduce binders like zeolite to act as porous carriers in the gut lumen, physically trapping leftover toxin particles. This sequestration intercepts toxins before they are reabsorbed by the liver through enterohepatic circulation, ensuring safe excretion. Once the environment is secured, we transition to Phase 2, identify. This requires utilizing precise qPCR GI map stool testing. We do not guess at the dysbiosis. This diagnostic tool extracts exact DNA counts, providing the specific ratios of beneficial bacteria versus pathogenic overgrowth, alongside concrete markers of mucosal integrity. With the exact bacterial deficits mapped, the support phase begins. The immediate biochemical priority is the glycine saturation strategy. The clinical intervention requires dosing 3 to 5 grams of pure glycine daily. Saturating the bloodstream at this volume biochemically crowds out the remaining glyphosate analogs. It guarantees the cellular machinery has the authentic amino acids required to execute precise protein synthesis. Simultaneously, we implement the 3-ferment rule, consuming 3 distinct live culture foods daily. This explicitly repopulates the highly sensitive keystone strains wiped out by the herbicide, particularly Akkermansia and Faecalibacterium prausnetzi. Systematically executing the reset, identify, and support phases seals the permeable gut barrier, stars out resistant pathogens, and rebuilds the biological foundation required for cellular energy signaling. As keystone gut microbes repopulate, they ferment dietary fiber into butyrate. This short-chain fatty acid serves as the ultimate metabolic fuel injector for human physiology. This chart breaks down the energy cascade. On the left side, we see the internal biochemical path. Butyrate travels from the colon to the cells, triggering the AMPK pathway. This activates PGC1-alpha, the master biological switch that orders the physical creation of brand new mitochondria. Now look at the right side of the chart. Specific wavelengths of therapeutic light penetrate dermal layers to physically strike cytochrome C oxidase, an enzyme inside the mitochondria. This bypasses the digestive system entirely, stimulating the electron transport chain and immediately spinning off ATP. But biological reality dictates that growing a new mitochondrial grid takes weeks, while a patient in systemic failure requires immediate energy to execute that cellular repair. To bridge this metabolic gap, we apply an external physical catalyst, clinical red and near-infrared light therapy. While butyrate slowly constructs a larger energy factory, targeted photobiomodulation manually forces your compromised engines to produce the vital energy necessary for immediate survival. Regenerating the microbiome and the cellular grid is a strict, time-dependent cellular process. During this first week, patients commonly experience the Herxheimer reaction, presenting as temporary fatigue and headaches. This early dip in energy is not a failure. This chart maps the exact physiological milestones you hit as your internal systems come back online. In the initial 24 to 72-hour window, Phase 1 begins. Biochemical sequestration takes hold, starving the remaining pathogens and initiating a rapid microbial shift. It's the predictable byproduct of pathogenic die-off in the gut. By day 30, Phase 2 begins, and the intestinal wall physically reseals, neutralizing systemic inflammation. Finally, Phase 3 extends to 120 days, birthing new mitochondria for peak cellular procession. To sustain this baseline during the Excel phase, clinical light therapy transitions to lifestyle habits, like utilizing early morning sun exposure to maintain circadian integrity. By systematically applying the RISE framework, saturating receptor sites with glycine, and leveraging targeted photobiomodulation, you completely bypass all environmental sabotage and successfully hardwire the human body back to a state of high-voltage vitality.